Good morning, everyone. So it's a pleasure today to have this video conference with Miltenyi company and also Aurélie Tchoghandjian, who has accepted to do this video conference. And today, Gaëlle Recher will moderate this session. She's a researcher and she's also a member of the steering committee of the GDR Imabio. So, Gaëlle? Your microphone is off. Sorry, maybe I did it.
No, but you did well. So good morning indeed, everyone. Thank you for attending this session. So as Virginie said, this session will start with an overview of different imaging platforms that are provided by the Miltenyi company. So the presentation will be by Eve Blancard. And then it will be followed by the talk provided by Aurélie Tchoghandjian. And then, if you are ready, we would be open to Q&A, so questions after both talks, if you don't mind. So write down if you have any question, and then we'll have an open session for addressing those. So maybe we can start with Eve, and then I will introduce Aurélie hereafter. Have you tried sharing your screen?
Eve, you can share.
Hello, I will share my screen now. So you should be able to see my screen.
Yes, fantastic.
Perfect. So I will start. So I'm very happy to be here today to present to you the solution we have with Miltenyi Biotec for the imaging, so in 3D and in spatial biology. So my name is Eve. I am a field application specialist for the Maxima, and I will be your speaker today. So Miltenyi Biotec, who are we? We are a global company that is present in 73 countries and distributed in 23 more. Close to 1/4 of our employees are part of the research and development department, and our headquarters is here in Europe, in Germany, in Bergisch Gladbach, for the one that know it. So Miltenyi Biotec developed a full workflow that goes from cell separation to isolation, cell culture, and analysis with our cytometer and our imaging solutions. And we are even now in the business of therapy with cell production that will directly bring to the patient. So the whole workflow that we develop and we use every day, we give it to you. So we bring it to you for you to use in your research and in translational research and in clinic. So today we will talk about imaging. So I will present to you in detail this part of our workflow. So we have two big pillars in the Miltenyi Biotec portfolio, with the 3D imaging with the ultra microscope Blaze platform, and the spatial biology that we do with the Maxima platform. So first, the 3D imaging with the ultra Blaze microscope. It's a platform that will allow you to go deep inside a full organism or an organ, and to have a targeted approach and to keep the 3D structure of your organ. How does it work? So to put it very simply, you extract your organ, you fix it, and then you will stain whatever target is interesting to you with 3D tested antibodies, directly coupled to fluorochrome. You will then clear your tissue, and you will image the whole organ or organism with a light sheet that will go through your organ and will allow you to have the full structure of your organism. The second pillar is the Maxima platform. That platform will allow you to do structural biology and spatial biology. So with the Maxima platform and the mixed technology that we developed, you go from a 5-plex on one slide to a 200+ marker on a single slide of tissue. So how does it work? With the Maxima imaging cycling staining, we stain the tissue, take a picture, and then erase the signal. And we do that cycling as many times as you theoretically want. And that's how we build up our panel. So to erase, we have two different methods. We can photo bleach the fluorochrome, or we'll go with targeted enzymatic digestion to remove the fluorochrome or the whole antibody from the tissue. So it's great to have 200+ marker on a tissue, but you need to be able to analyze it. So we developed a software, MaxIQ-View, that has been tailor-made for the Maxima and the Maxima data that will allow you to do all the key features that you need to understand and dissect the biology of your tissue. So you can do segmentation directly inside the tissue, inside the software, gating, you do spatial analysis, you can do clustering and dimension reduction with TISMI and UMAPs directly built in. So the software has been developed with the Maxima, is very easy to use and easy to start with, and you get directly ready to publish data and ready to present data. So that's the two pillars of the Miltenyi imaging system. But if you want to go even further and be at cutting edge at the farthest you can be in development and technology, you can combine the two systems. You can image a whole organ in 3D and then re-embed and cut that tissue. And that will allow you to have the information of the 3D and targeted approach, for example, localize your tumor and your metastasis. And then when you cut, you will have the advantages of the multiomics and the multi-parameter. So that's just an example what we did on the mouse cerebellum, that we targeted a location that was interesting to us, and we did panel with different staining, and you can see on the right side the detail you can have on your slide. So thank you for your attention, and I can't wait to hear Aurélie that used those technologies.
Well, thank you very much, Yves. So indeed, we are welcoming Aurélie Tchoghandjian today. So she's a senior honoris researcher at La Timone in Marseille. She co-leads the team GlioME and Microenvironnement. And since her PhD, actually, she was interested in GlioME systems and studying them from the cellular point of view, and also using imaging techniques. So she has been she has been co-leading our team since 2022, and also started a network called Petra. I don't know whether she planned to tell us a little bit about that, but it's Petra for preclinical and translational research in neuro-oncology. So maybe actually it's better to hear her directly talking about what she does there in Marseille and showing us, I'm pretty sure, beautiful pictures and movies with lots of insights. And so Aurélie, the floor is yours.
Thank you, Gaëlle, for this introduction. So today I will present you how in the lab, in the team, we are using light sheet, so 3D imaging, and multiplexed spatial proteomics, so mixed by using the Maxima platform, to study glioblastoma microenvironment. So sorry, I don't know. I cannot. Okay. Our team, in the GlioME team, we aim at discovering new targets and understand their role and their function to design innovative treatment. So our team is composed of researchers, engineers, pharmacists, clinicians, pathologists, and we start from the patient and go back to the patient. So we have a bed-to-bench and a bench-to-bed approach. And so we are working on brain tumors, on glioblastoma. So brain tumors are rare tumors, rare disease, 1% of all cancer. But glioblastomas are the most frequent and aggressive brain tumors in adults. So I can maybe just take a pointer. So how you can see here, the tumors can be really huge with the infiltrative zone. And here it's the core of the tumor. And so the standard treatment is chemotherapy followed by radiotherapy, but still the median of survival is very low and tumors always recur. So how is so complicated to treat these tumors? Because this protocol of treatment is quite old. We are using it since 20 years now. So why it's so complicated to treat these tumors? So it's a multifactorial tumor, and the tumor microenvironment play a key role in tumor growth and treatment resistance. And the tumor microenvironment is highly heterogeneous and highly complex, composed, for example, of several types of cells, some glial cells, so brain-resident cells, but also blood-derived cells as lymphocytes, neutrophils, dendritic cells, and TAMs. So the tumor-associated macrophages, so macrophages and macroglia cells. And all these cells of the microenvironment constitute up to 1/2 of the tumor. So these tumor-associated macrophages are the most abundant cells of the microenvironment within glioblastoma. And they play a central role in tumor progression, immunosuppression, and cerebral edema. So finding way to reprogram these tumor-associated macrophages toward immune supporting cells is one current challenges in the field. So what are these tumor-associated macrophages? So they are composed of macroglia cells and monocyte-derived cells. So macroglia derived from the yolk sac, and they are present in the brain parenchyma, and they are able to invade and infiltrate tumors. They express TMEM119, and they are highly branched when they are in basal state, and they become amoeboid when they become activated. So the monocytes are recruited to the tumors, and here you can see all these red dots in the bloodstream. So they are going inside the bloodstream to infiltrate tumors, and they are becoming monocyte-derived cells or macrophages. So how to reprogram tumor-associated macrophages so to restore a functional immunity in glioblastoma? So in the team, we are using small molecules, so SMAC mimetics. The first role is a pro-apoptotic role, and they are antagonists of inhibitor of apoptosis proteins. And what we show, they are capable to differentiate stem cells to trigger apoptosis, and also to trigger apoptosis and to increase mice survival. But what we know also about these proteins and these small molecules, they are involved in NF-kappa B signaling and TNF-alpha signaling. So they are involved in inflammatory pathways. So what could be the role on tumor-associated macrophages remodeling? So this was the question that Emmanuel Snazelfazi tried to answer during his PhD. And to do this, we used the ultramicroscope blaze to perform light sheet imaging. And so he developed the technique in the lab and also the clearing processes. So to know about these cells, their densities and their spatial distributions, we used light sheet imaging. And so here it's half of the brain with a tumor. So the tumor is in red, the immune cells CD45 positives are in yellow, and the vessels are in purple. And we use the combination of primary and secondary antibodies to obtain these results. And the images were analyzed using Imari software. So by using this tech, then, so after the image, we have to process these images. And so there is a step of segmentation. And so to segment the tumor, we decided just to focus on the bulk of the tumor, because these tumors are highly invasive, but to have some insight about the size of the tumors. So we segment the tumors, we segment the vessels and the immune cells, and then we are able to quantify the immune cells only inside the tumors, or the vascular densities present only inside the tumors. So one dot is one immune cells. And so by doing this, we were able to quantify the density of immune cells at different times during tumor growth. So we sacrificed mice at different timing after grafting of tumor cells. So we grafted tumor cells, and then the tumor started to grow, and we started the treatment 1 week after the graft of the tumor cells. And then 2 weeks, 3 weeks, and 4 weeks after, we decided to take out the brains and then to clear them and to make Woolman stainings, and then all the process that I just showed you. And we analyzed and quantified the densities of immune cells inside tumors. So you can see that quite early in tumor development, 2 weeks after tumor graft, we have immune cells, and we have much more immune cells with the treatment, so with the SMAC mimetic treatment. And this is quite stable during tumor growth. We obtained the same results after 3 weeks and after 4 weeks of treatment. So the treatment increased immune cell quantity inside tumors. So we wanted to know also a little bit more about the distribution of the cells, because what we know is that in solid tumors, immune cells are often located at the borders, at the periphery. It's difficult for them to infiltrate inside the tumor core. So we wanted to know if with the treatment, we were able, in addition to density, to promote infiltration of these immune cells. And so we just took the same images of the brains, and then we separated the tumors in layers based on the center of the tumor. So the center is here in purple, and then we make 4 layers. So the outside layer is far away from the center. And from this, we could determine where are localized the immune cells, if they are inside, in the middle, or at the periphery of the tumor. And so here you have the heat map, so representing the shortest distance to surface. So we measured the tumor center and then reported to the tumor border. And using this color-coded, we were able to classify the immune cells. So here is in vehicle conditions, and here is with the treatment. And what we can see is that the immune cells were able to infiltrate deeper these tumors. So the treatment increased tumor immune cell density, but also infiltration inside the tumor core. Then we wanted also to know what about the relationship with the vessels, because we know that immune cells can cluster around vessels, and then we wanted to know if they could be relocated from perivascular spaces to intratumoral areas, and this needed to increase the resolution, and we needed a single cell resolution. So we imaged again the same brains, but at a higher resolution. And we can then nicely have a single cell resolution, and you can see the different morphology of the immune cells and of the vessels as well. So by using these images, we were able to make another heat map. So each dot is an immune cell, and when they are red, they are far from the vessels, and when they are blue, they are close to the vessels. And we quantify this, and what we obtain is that with the treatment, we have more red than with the vehicle condition. So we have an infiltration from perivascular areas to the tumor parenchyma. So until now, we analyzed the immune cells, and we know that these tumor-associated macrophages are the most abundant. And then we decided to focus on microglia because they are the brain-resident immune cells. And so we set up also the clearing protocol and light sheet imaging to see these microglia inside tumors. And here they mainly have an amoeboid shape, so it's because they invaded the tumors, they are inside the tumors. So we did the same than for the immune cells. So we determined the densities inside tumors. So here on the upper scheme, you can see that, so this is the vehicle and this is the treated tumors, and we have a decrease of the treatment, and this decrease of the treatment means that the tumors so have less TMEM119 cells, and we know that this marker is a marker of basal microglia, and when microglia become activated, the cells lose this marker. So here we have a decrease of this marker with the treatment. And if we have a look also to the morphology, we can see that the morphology of the cells also change, reflecting probably changes in function of the cells. So then we also analyzed the location related to the vessels, as we did for the total immune cells, so the same heat map and the same colors. And here again, we can potential so this perivascular to intratumoral infiltration of these microglia cells. So to study the functional role of this microglia, then we switched to a human-derived model, and we co-cultured microglia with total tumor, so tumoroids. So what is this model? We used both techniques, so Maxima and light sheet imaging, to analyze these co-cultures. So what are these tumoroids? So we take a tumor from patients, and then we sorted the microglia on one side, and we cutted the tumor in small pieces on the other side. And then we platted the microglia cells, and we make a co-culture with these pieces of tumor that we call tumoroids, and we put these tumoroids on the top of the microglia cells. But first, we needed to characterize these tumoroids, so these small pieces of tumor, to be sure that we preserved all the glioblastoma characteristic, and that this tumoroid preserved the same characteristic than the patients. So these are the small pieces cut in culture, and 1 week later, they form these small structures, and they become round. And we can find the same histopathological characteristic than found in human glioblastoma, so necrosis and angiogenesis. So to better characterize them, we performed Maxima. And so we froze these tumoroids, and then we cut them. We put them on the small slides required for Maxima technique. And then we selected 20 markers. 20 markers that they are specific for human and also specific for PFA fixation. And we perform so this Maxima technique. So it was quite fast because it was only one region of interest and 20 antibodies. And these are the results that we obtained. So I present you just some stainings. But what we can see, and it was very surprising for us, it's that inside the tumoroids, it's regionalized. So we can see very specific areas corresponding to specific cell subtypes. So for example, here we have the GD3 positive cells, so a subtype, a type of tumor cells, and they are here. And we have beta-3 tubulin, so for immature neuron, and they are here. We have neurons, MAP2, they are there. And we have also here, it's the microglia cells, so they are here, the hypoxic region, and it was more or less the same for each type of cells. And so we were quite happy about these results because we did not really expect such an organization inside these tumoroids. And it's highly precious, these results, because we can do several and a lot of staining on this tissue. And using human tissue, actually, it's very important that we can maximize the use of this tissue because they are rare and we cannot have as much as we can. So this is a technique which is totally compatible with the use of human tissue. And we also wanted to know whether this tumoroid model could be a functional model before to go on with our experiment with the co-culture of microglia, and if they are sensitive to drug and if they are capable to synergize with other drug. And so we treated the tumoroids with one treatment. Oh, sorry, I miss the legend here. So this is the SMAC mimetic. This is the temozolomide, so the standard chemotherapy. And this is a combination of both. And we use PKH 26 to have access to the volume of the tumoroid. And we use a rapporteur, a fluorescent rapporteur of K-specific cleavage for apoptosis. And we performed the Woolman staining for Ki67, so to see proliferation. And for this, we used the brain, the clearing kit of Miltenyi, and this is also an antibody directly coupled to the fluorophore sold by Miltenyi Biotec. And what we can see is our treatment promotes trigger apoptosis, decrease proliferation. Temozolomide, we don't really have apoptosis, but decrease a little bit proliferation. But both, we really gain more apoptosis, and we can also nicely decrease proliferation, and we quantify this by using Imaris software. So this also 3D technique worked quite well on these tumoroids. So we applied it to the co-culture of microglia and tumoroids. So here you can see only the tumoroids. We cannot see the monolayer, sorry, of microglia because we took out only the tumoroids after the co-culture. So we platted the microglia, we treated the microglia with the SMAC mimetics, and then we removed the treatment and we put the tumoroids. And then we wanted to see if we obtained the same results with or without our drug and with or without microglia. So what we can see is no much difference with or without microglia. Sorry, with the treatment. This is without microglia. So the treatment has no effect because the dose that we use cannot trigger apoptosis. But then when we add microglia, we cannot really promote, there is no effect on apoptosis, but we promote apoptosis with microglia. And this is what you can see. So the tumoroid alone, the tumoroid without microglia, the tumoroid with microglia but without the treatment, and with microglia and with the treatment. So that means that we have an impact on apoptosis only with microglia and with the SMAC mimetics. So microglia is a key actor of the SMAC mimetic response in glioblastoma. So then we wanted to know if MLIP can be the target of SMAC mimetic. So what we show previously is that this SMAC mimetic could trigger 4 inhibitors of apoptosis protein, including MLIP. And what we showed in cohorts of human glioblastoma is that only MLIP is a factor of bad prognosis in these tumors. So we tested this in 2 cohorts, and here is progression-free survival. So it means at relapse, and here it's overall survival. So when the patient died. And we quantified the expression of MLIP. And what we found is that when MLIP is strongly expressed, it's bad for the patient. The patient died earlier. And we got the same results for the PFS or for the OS. So we wanted to know if MLIP could be involved in this SMAC mimetic response. So this is a work of Raphaël Bergès and Soline Toutain in the team. And so what they did, so they perform cell lines, glioma cell lines without MLIP, so by shRNA technology, and they grafted these cells with or without MLIP in the brain of immunocompetent mice, and they treated or not the mice. And what we can see is that the cells expressing MLIP rapidly derived tumors. And when we treat with SMAC mimetic, we delayed the tumor growth and we increased the survival. This we confirmed what we had previously on immunodeficient mice. And what we obtained is when we removed MLIP, so in these SH MLIP cells, we increased much more the survival of the mice, but then the increase that we got with the treatment was less. So this means that MLIP is probably highly important during tumor growth. So to investigate its role, knowing that this protein can be involved in the microenvironment, in the tumor-associated macrophages, in also the distribution of the immune cells inside the tumor, we decided to use the Maxima platform to characterize the microenvironment associated to MLIP down expression in the tumors. And so all these Maxima technique are doing in collaboration with the ICE platform at CRCM in Marseille. And for this mouse experiments, we used a panel of 56 antibodies. 53 are from Miltenyi Biotec, and we use 3 others from other company. And this platform so gives us also some flexibility because we can also use antibodies which are not the antibodies of the company, but we can couple the antibodies to fluorophore or to use primary and secondary antibodies. And this works actually quite well. And we used antibodies recommended only for PFA fixation because we used so far only frozen mouse tissue. And so I show you the results of the test. We did not have the results for the real experiment so far. But we have panels for tumor cells, and here you can see GG3 for stem-like cells, SOX2 for tumor cells, CG44, and Ki67 for proliferation. And this is the merge of all the staining. And we also have a panel for vessels, so CG105 for angiogenic vessels. And this is a merge of all of these vessels. So we can see that, for example, alpha SMA is not homogeneously distributed in the tumor. And we have also a panel for macrophages, so the TMEM119, so for the microglia cells, and we can see, and this is described in the literature, that these microglia cells are found at the periphery of the tumor. And this is MHA2, so the cells with antigen presentation capability. And this is galectin-3, which are expressed by blood-derived cells. And this is a merge of all these markers. And we can see that we have clusters of cells here of TMM, so macroglia cells. And we see the normal microglia, the branch microglia in the parenchyma. And these are other markers for macrophages, but what we can see, they are organized in clusters, mainly as we saw for the tumoroids, so that the cells of the microenvironment, they are specific areas. And this is also the merge of all these markers. And a panel for lymphocytes. So the lymphocytes are quite few in glioblastoma tumors. So I made some a zoom on these cells. So they are present, they are clustered. Again, they are found in specific areas, but they are not a lot. So that's why it's important to have this really nice single cell resolution. So, and a panel for neural cells, for oligodendrocytes, astrocytes, and we can see here that astrocytes, they make also a kind of they are really located around the tumor, and they make this kind of layer around the tumors, and this was not really expected as well. And so for the analysis, so we can play with several parameters, with the intensity, co-expression. So the software is quite easy, is really friendly to use. And I'm not a bioinformatician, and I can use this software. So it's quite easy to use. And what it's nice, it's if you are using, for example, flow cytometry, you can apply your flow cytometry gating strategy. And what we did actually, because we wanted to phenotype the immune cells, and so as I explained you, there are quite a lot of different subpopulations in glioblastomas. So we started from our gating strategy that we use for flow cytometry, and we try to apply this for the mixed technique. And so for the analysis, then it's easy because we have all the workflow for the analysis, and it's much more easy to think about that before than, okay, this marker could be nice to see, this one could be nice, and then at the end we don't know how to correlate all these markers. And so that's what we do. So this is our workflow of analysis, just to show you that we can really analyze a lot of different cell subtypes. We can really subclass all these cells, and we can quantify, we can perform a UMAP and so on. But okay, we are ready now to perform our the real experiment, and so our workflow of analysis is ready. And so in conclusion, so the mix experiment is set up, we are ready, and we are going to analyze tumors at several times during tumor growth to understand what happened with or without MLAP expression, to understand how the microenvironment is reprogrammed with or without MLAP. And after this mixed approach, we will combine it with 3D approach, and we expect to find few cell subtypes that we can go deeper so in 3D to see what happens between both populations. And so these techniques need time to set up. It's an investment, so time, money for the analysis, but then when they are set up, they can bring a lot of results and they can be used for several applications, and they are highly complementary techniques. So I thank all the GlioME team, and also, as Gaël said, I coordinate the Petra network, which is dedicated to preclinical and translational research in neuro-oncology. So we provide platforms to do this, and it's in this platform that we have these models of tumoroids, of glioma experimentation, and so on. So I thanks also to the collaborators involved in this project, the ICEP platform and the INFIM platform for the light sheet, all our financiers, and of course Miltenyi Biotec for the support and for the help. And I thank you for your attention.
Thank you very much, Aurélie. As expected, very nice and beautiful images, movies, but also a great science. So if any of you have any questions, so either I see that Leslie already raised her hand, so turn on your microphone, and or if you're shy, and if possibly the video so that we can see your face. But if you're shy, maybe you can just type on the discussion forum, and we will read it out loud.
I just remind you that the video is recorded, so if your camera is on,
yes,
it will be on the video. So if you prefer to not be recorded on the video, please switch off the camera. Thanks.
Who wish to start? Either questions for Yves or for Aurélie? Are there any? Alessandro.
Yes, here I am. Hello, thank you very much for this great presentation. I had some question about hypoxia in the tumor, and is there some link with some markers that you could evidence?
Yes, so that's why we include GLUT1 in the mix panel. So glioblastoma are necrotic tumors, so they are areas of hypoxia. And what we saw, it's the drug that we are using, so this MAC mimetics, depending on the level of oxygen, can be either can trigger either apoptosis or can trigger the differentiation of the stem cells. So that's why it was important in our condition to have an insight about what happens about hypoxia. And so I and
tumor cells, but about immune cells, could you also see some correlation between?
So far we did not. What is complicated in 3D, in the light sheet imaging, we did not investigate hypoxia so far. We did not try to use one of the hypoxic rapporteur or level of oxygen rapporteur if it can be suitable with the clearing technique and the light sheet imaging. So we did not go into this direction, but that could be highly interesting. But we would like to see stem cells and immune cells and hypoxia. And we are just thinking about to use the good model to see all the different partners.
Okay, and if I can go on, did you have issue about cell segmentation in your to see for individual cells?
So which technique for the light sheets?
Yes.
Yes.
And depending on the objective and.
No, actually, at 4x, the single cell resolution, it's quite good. We used 2.5 zoom. It was good enough. We did not need to go to a 12x objective. We tried. It works, but the staining need to be very, very good and very bright. This what we concluded actually. But so if you have a very good staining, could be very nice to go to 12x.
Okay. A small last question about microglia cell lines. Do you have some comments on which one to use, or I don't see many in the literature?
Yeah, so actually here we used microglia cells that we sorted from human samples, but we could not keep them for a long time in culture because they are quite sensitive. So human microglia cell lines are not very reliable. We cannot really use them. They're not very good cell lines. So we use mouse microglia cells. Or a lot of people use also microglia derived from iPSCs.
Okay. Thank you very much.
You're welcome.
Thank you, Alessandro, for your questions. So Eve already replied online, but there were a couple of questions in the chat from Yuan Shu. Sorry for the pronunciation. So he or she was wondering whether the solution of the MAX is for the clearing provided with the MAX system is toxic, and so Eve answered that no, but actually I had also questions to press on that question. So is this based on the published techniques, or is it patented and very secret? And then is it more based on the lipid removal, or just trying to match the index by exchanging the water by something with a higher index somehow?
So I'm not a specialist of this technique. It's Christian Feuillet that is a specialist. I think you know him. But from what I know, it's lipid removal that will allow you to clear your tissue like that.
Okay.
Yeah.
Okay. So then maybe more than the techniques from Clarity with the SDS solubilization of lipids or something like that then? Okay,
Yeah.
we'll ask Christian.
Yeah, I'm so sorry, I can't help you with that.
No problem. So the second question was whether how you erase the signal in the system, and so maybe you can say it aloud and explain.
Yeah, maybe I can say it. So we use photo bleaching. So we basically turn on a very strong LED that will denaturate the fluorochrome, or we use enzymatic digestion. So we have recombinant antibody where we place the sequence that we come to target with a specific enzyme that will cleave this sequence and release the fluorochrome, or release the whole complex, antibody and fluorochrome, from the tissue, so you won't have any more signal.
And so just by curiosity, you flow some solution. I saw the bottles on the side of the machine. So the sample stays in place, and then there is a system of flowing the different solutions step by step. Is that it?
Yes, basically. So the bottles you saw in the side of the machine was to wash the sample, dilute antibodies, and wash the whole maxima. but when you do enzymatic digestion, so we provide you some vial in a kit that you put directly inside the maxima, and the device will dilute the solution, apply it to your sample, and incubate it the right amount of time, and then remove it. So it's a fully automated robot. And once you have placed your sample, your antibodies, you don't have to touch anything anymore.
Okay, thank you. Are there any more questions from the audience? Anybody raise this or her hand? So we had one more from the same person who's asking what's the distance between CD45 positive cells from the vessel? So I assume that this question is for Aurélie.
Yeah, how is it important? So actually, it's because immune cells have been described to be at tumor borders. Tumor borders could be in perivascular areas or at the periphery of the tumor. And that's what we wanted to know, because there are a lot of immune cells in glioblastoma. Half of the tumor mass is more or less immune cells. But if the immune cells stay outside in the tumor borders areas, then they cannot they cannot fight tumor cells. So that's why it's important also to promote the infiltration inside the tumor. That's why it was important to understand what happens related to the tumor borders, but also to the perivascular areas. And then I see another question. So for the blood vessels, we are using the protocol of Nicolas Régnier, published in Cells it was 2020, I guess. It's a combination of 3 markers: alpha SMA, CD31, and podocalixin.
For the vessels?
For the vessels, yes. So like this, we can stain the big and the very small vessels.
Oh, may I ask why not CD31?
Yeah, because you cannot cover all the type of vessels, and with only CD31, we cannot map all of them. So, but alpha SMA and CD31 is already good, but it's also could be better in some condition to add podocalixin.
Mm-hmm. Okay. Virginie, you raised your hand.
Yes, just a practical question, but you said that it takes time to develop the pipeline with all your antibodies. So how many weeks or months it took to have your pipeline with
Light
6 antibodies?
For the mix for the maxima?
Yeah.
Yeah, so we started to think about the panel, I think more or less 1 year ago, because it's a lot of antibodies, and then we want to select the best. You want to use all of them, but you cannot, so you have to select somehow. So it takes time to maybe a few months to adjust the panel, and then when you find the antibodies, you also have to find the good way to process your sample. And then when this is done, you started with to try the panel of antibodies. For us, we were quite lucky because most of us worked at first, just like 8 or 7 did not work properly. So we increased the concentration for some of them, and then some worked quite well. Some did not work very well, and we decided to remove them and to try some others, because there are different clones also. So sometimes you have to test one, but it's not working, and you can try then another one. But also Eve and all the team is helping in the different steps, and it's a lot of discussions also. And we did this on the ISEP platform, and also we had a lot of discussions with the platform. And now we, so I think, had some issues with the samples and so on, because we freeze the samples before to fix them, and we realized that before to cut the samples, we have to cut the samples just a few days before the technique. And we did not realize this earlier, and yeah, this just came out, so that's why we did not have time to. We did not have yet the real experiment, but we have 5 samples. We also have to find a way to put the 5 samples on one slide, and these are small things, but each time it's small settings, and okay, one after the other. It's a little bit of time, but then when it's working, I think when we find a good workflow, then it's fine. But for the analysis, actually, it was quite fast. It's maybe not so complicated, because we knew before what we want. We already have the gating strategy before to start. So I think that's why it's maybe faster, this part.
Thank you.
Related to that, actually, I'm curious whether so it's a question for both of you, actually, even Aurélie. So do you rely on a huge community in order to exchange protocols and so on? I mean, maybe nationally or internationally, are there other in France, because this is highly technical, and maybe people would have troubleshooted before, and so since the machine is on a platform, and are there any user network available somehow?
So that's something we are trying to develop, but we are at the beginning for the Maxima. And, the first one we installed was in Marseille at the ICEP platform, but now there is 3 more installed in Paris. So we are very happy about that. And we are trying to have meetings every year in Miltenyi, where we bring in the users of the Maxima and the Blaze, to have them talk to each other, to have that kind of network to develop. But yeah, that's something we encourage, and I don't know how structured it is yet.
Okay.
Yeah, and also, for example, for the tumoroids, so it was also a lot of settings to understand how to put this tumoroid on the slide, how to keep them in the slide, because the first experiment, when we use the Maxima, we have nothing on the slide left, because all the tumoroids were removed by the cleaning and so on. So we also have to adapt how we proceed the tumoroid, how we freeze them, how we fix them, how we put them on the slide. So there are also some, so to understand how to proceed with the sample, it's also a little bit complicated. I think for FFPE sample, it's easier maybe. For sample fixed formalin, for this kind of sample, maybe I think it's easier than for frozen samples.
Yeah, we have ready-to-use protocol for very straightforward things, but Aurélie was doing something very cutting edge, and we didn't have that much experience with it. So I think she will be able to help other users in the future. But yeah, it's always someone has to start.
Okay. Regarding to that, actually, what is the vessel? Is that consumable provided by you? Is it because I've just read quickly that your objectives, they have a very long walking in distance, so that they can go through 1 millimeter slides. But I assume that, considering that you have to wash, that the volume has to be minimized and everything, you have a specific vessel for doing that?
So we use regular macroscopy slides that everybody has in the lab, Superfrost Plus or anything you use, so 1 millimeter thick slide. And we build little chambers around that slide, so that the consumable that is provided by Miltenyi, specially designed for the Maxima. But it's pretty much a chamber with some rubber on the side, not to have liquid going everywhere, but yeah, it's consumable. We make.
So it's a sort of a silicone gasket or something like that.
Yeah, something like that, like a casket that you put on top of your slide, and you kind of lock it, and then you have a well around your microscopy slide. Okay.
Because just I'm used to hack systems usually and think about the different things, and so Aurélie mentioned that actually ours tumoroids were washed out, but if there were a way eventually to get them to stick to the slide, that would be feasible to emit them theoretically and to do the whole protocol.
Yeah, once the problem with the tumoroid was even before they got to the Maxima. That was
the whole processing of the...
Yes, the whole process. They would get washed away when you wash your DAPI or anything you are washing. Once your tissue is sticking to your microscopy slide, you are fine to build your chamber around, and the tissue should resist the whole process. We went to 400 cycles of washing and staining, and the tissue stuck to the slide. So it's a very gentle flow we create, because we don't want to have the tissue floating around and to have the epitope removed or altered in any way.
Okay. Very specific. And I assume that the volumes also that you use are very tiny, so that you don't waste.
Yeah, we have different sizes in chambers. So we have a chamber that will do the whole slide, that 1 millimeter volume, so that's quite high, but you can divide this chamber. So we have a small chamber that is 250 microliter. So you prepare your antibody for this volume.
Okay. Thank you. Are there any more questions in the audience? People are very shy, maybe because we speak too much, but people, raise your hand. No more? Okay, I may just have a last one about this machine, actually. So it was mentioned that it's a wide field microscope. Are there any further developments in order to turn it into a confocal microscope somehow?
Yeah, that's the next step of the Maxima, but it's not ready and it's not announced or anything. For the moment, we are still developing on the wide field microscope, and we can already do a lot, but yeah, it would be very nice to have that.
Sure. Thank you. What do you think, Virginie? If we have no more questions from the audience? Maybe?
Yeah, I think it's time.
Yes.
To close the session. So thank you very much, Yves and Aurélie, for this session. Thanks also to Gaël to moderate and animate this session. And so the next video conference of this series, organized by the GDR Imabio, is the 7th of May. And it will be done with T-Light bioaxial about the phase imaging. So hope to see you there. So thanks a lot to all of you.
Thank you for the invitation.
Thank you.
Bye.
Bye.